Hello and welcome to today's video lecture on chikungunya, a viral illness transmitted to humans by infected mosquitoes. Let us delve into the intricacies of this often misunderstood disease, exploring its origins, symptoms, treatment options, and preventative measures. Now, let us look into the historical perspective of chikungunya. Although referred to as chikungunya, this fever has nothing to do with chickens. Chikungunya fever is an arboviral disease that is caused by chikungunya virus and is transmitted by mosquitoes. Chikungunya is a key Makonde word meaning that which bends up and refers to the contorted posture of infected patients suffering from severe joint pain. The first cases of a chikungunya-like illness in humans were recorded in 1823 in Zanzibar, Africa. Similar epidemic occurred in St. Thomas Island in the Caribbean during the years 1827 and 1828. In 1952, episodes of rheumatic fever affected several people in Tanzania, where for the first time chikungunya virus was isolated, identified, and characterized as an arbovirus. The most remarkable outbreak occurred between 2005 and 2006 in La Reunion Islands in the Indian Ocean. During this epidemic, over one-third of island's population was infected. Around 2,60,000 people with an average of 40,000 new cases per week were recorded and 284 deaths occurred. Worldwide distribution of chikungunya virus. The disease occurs in Africa and Asia, although imported cases have been recorded in the World Health Organization's European region and the region of Americas. In 2023, as of 31st of December, approximately 5 lakh chikungunya viral disease cases and over 400 deaths have been reported worldwide. Genotypes of the chikungunya virus. Currently, 3 genotypes have been identified based on genealogical relationships within the chikungunya virus. This phylogenetic tree represents the important genotypes of the chikungunya virus. The genotypes are of 3 main types: the East Central South Africa strain, West Africa strain, and the Asian genotype. Within the East Central South Africa genotype, there is a lineage that is considered very important due to its biological characteristics. This lineage is called the Indian Ocean lineage. Transmission cycle of the chikungunya virus. A. Sylvatic cycle. The virus is transmitted between the non-human primates with the participation of many species of forest-dwelling mosquitoes of the Aedes genus. The rural cycle. During heavy rainfalls, the forest-dwelling mosquitoes enter nearby villages and can transmit the infection to humans. During the spread of infection, the domesticated mosquitoes Aedes aegypti becomes infected and can participate in the chikungunya transmission. The urban cycle. The human migration towards large urban centers can translocate the infection, where the transmission is performed by urban mosquitoes Aedes aegypti and Aedes albopictus. The sylvatic cycle of chikungunya transmission has been described only in Africa, while in Asia, the transmission is maintained only between humans. That is, only urban cycle, without the participation of reservoirs. After the introduction of chikungunya virus in the Americas, the viral transmission is maintained exclusively in the urban cycles. The ability of arthropods to transmit pathogens depends on intrinsic and extrinsic factors and is expressed in two terms. Vector competence, that is, the ability of a vector to become infected and transmit after the pathogen is ingested during a blood meal. It is often regulated for arboviruses at the level of midgut infection and vectorial capacity, that is, the number of infective bites arising from an infected host. Host-seeking female mosquitoes are infected after feeding on a viremic animal. Chikungunya virus first replicates in the midgut and then enters the hemocoel before disseminating to the salivary glands. The midgut basal lamina reorganization during blood digestion mediates this dissemination process. The extrinsic incubation period is generally 2 to 5 days, suggesting that even vector populations with poor daily adult survival can transmit effectively. Females with disseminated virus in their salivary glands can transmit by injecting infectious saliva into a naive host during a subsequent blood meal, leading to horizontal transmission. Aedes aegypti feeding is often interrupted when it is disturbed during blood feeding, and it may complete the meal on one or more hosts in the vicinity. This can lead to this highly anthropophilic species to feed on multiple persons daily, increasing the risk of chikungunya virus infection and transmission to multiple hosts, greatly enhancing vectorial capacity. Vector competence of Aedes aegypti and Aedes albopictus show variation according to the geographical origin of the mosquito population and chikungunya virus strain. Once infectious, the mosquito is believed to be capable of transmitting the virus for the rest of its life. Factors attributed to the transmission of the chikungunya virus. There are several factors that define transmission. Climate can affect the transmission dynamics, geographic spread, and re-emergence of vector-borne diseases through multiple pathways, including direct effects on the pathogen, the vector, non-human hosts, and humans. In addition to having direct effects on individual species, climate change can alter entire ecosystem habitats, including urban habitats, in which vectors or non-human hosts may thrive or fail. Because arthropods and other vectors are ectotherms meaning animal dependent on external sources of body heat, it is expected that vector abundance and survival and feeding activity will increase with increasing temperature, as will the rate of development of the pathogen within the vector. As the earth warms, the concerns are that mosquito and virus will spread to higher latitudes and altitudes, that incidence will increase and the transmission season will lengthen in some endemic areas. The relationship between precipitation, that is rainfall, and vector abundance is also complex and context specific. Increased precipitation could provide more vector breeding sites. However, drought could also provide more breeding sites due to an increase in the use of containers for rainwater collection and storage, which serve as primary breeding sites for Aedes aegypti. Vector abundance could also be influenced by how often water storage containers are emptied, and this is in turn determined by climate and the availability of running water. Vector abundance is also influenced by ecosystem change, which could degrade or enhance vector habitats and species competition, or it could increase or reduce the abundance of vector predators or vector pathogens. Human population displacement caused by climate change can spread to non-endemic localities through travel, trade, or migration. Mosquito hotspots within a city can be particularly problematic following rapid increases in urbanization and unplanned growth. These factors may give rise to a lack of sanitation and infrastructure with little clean water, sewage, or waste management, and therefore abundant adult mosquitoes. Scientists say that dry weather makes mosquitoes thirsty, and when they become dehydrated, they want to feed on blood more than often. Globalization has a profound impact on the flow of people, and for domesticated Aedes aegypti species, human transport of man-made containers contaminated with diaposing eggs and adult mosquitoes has facilitated their dispersal. Travelers from no-risk areas are vulnerable when traveling to areas with virus circulation, particularly during an outbreak, because most of them are immunologically naive. Infected travelers who return home to areas where Chikungunya virus competent mosquitoes are present could give rise to local clusters or outbreaks. Illness in individuals from poor backgrounds can have serious consequences, such as reduced productivity at the individual and community level, malnutrition, other infections, socio-economic instability, and exacerbation of poverty. Virion structure of the Chikungunya virus. Chikungunya virus is a member of the Old World Semliki virus group of arthritogenic alpha viruses within the Togaviridae family. It is an enveloped plus strand RNA virus with the icosahedral symmetry. The virion is 70 nanometer in diameter and is composed of repeating units of E1 and E2 transmembrane glycoproteins. Around 240 heterodimers of E2 and E1 are arranged as trimeric spikes on its surface. The capsid C is a host-derived lipid bilayer and consists of a single molecule of genomic RNA. Now, let us look into the genomic organization of the Chikungunya virus. The genome of Chikungunya virus consists of a positive sense single-stranded RNA molecule, which is approximately 11.8 kilobases in length with a 5-methyl guanosine cap and a 3' polyadenylate tail. The genome is divided into two ORFs, ORF1 and ORF2. ORF1 and ORF2 encode for non-structural proteins and structural proteins. The expression of ORF1 is controlled by genomic promoter located in the 5' untranslated region, and the expression of ORF2 is regulated by an internal promoter. The genomic RNA is employed as a template for synthesis of new viral genome for Chikungunya virion assembly. The subgenomic RNA is used as a template for production of viral structural proteins. The known functions of individual Chikungunya viral proteins are as follows. The gene NSP1 codes for non-structural protein of the size 535 amino acids. It displays methyltransferase and guanyl transferase activity that caps the viral RNA. It also serves as a sole membrane anchor for the replicase complex. The non-structural protein 2, NSP2, is of the size of 798 amino acids. It displays N-terminal, antipase, helicase, and RNA triphosphatase activities. The C-terminal cysteine protease activity is responsible for processing of the non-structural polyprotein of the virus. NSP3, the non-structural protein P3, has a size of 530 amino acids. It is a phosphoprotein with unknown functions, but it is important for minor strand synthesis. It contains macrodomain and the SH3 binding domains. It likely interacts with host proteins. NSP4, non-structural protein 4 gene codes for a protein consisting of 611 amino acids. It displays RNA-dependent RNA polymerase activity and also putative terminal transferase activity. Now, coming to the structural proteins. The capsid protein C is of the size of 261 amino acids. It encapsulates the genomic RNA to form a nucleocapsid core. The carboxyl domain is an autocatalytic serine protease. The protein E2. The protein E2 is of the size of 487 amino acids. It is an intermediate composed of E3 and E2, and it is cleaved by the host furin protease. The protein E3 is a short protein consisting of only 64 amino acids. Its N-terminal domain is the uncleaved leader peptide for E2, and it may shield the fusion peptide in E1 during egress. E2. The E2 gene is of the size of 423 amino acids, and it mediates binding to receptors and attachment factors on the cell membrane. It is a major target of neutralizing antibodies. The protein 6K is a short protein of 61 amino acids. It is a leader peptide for E1. It also serves as a putative ion channel and may enhance particle release. The protein E1 is of the size of 439 amino acids. It is a type 2 fusion protein. It mediates the fusion of viral envelope and cell membrane via fusion protein. Now, let us look into the replication of the Chikungunya virions in mammalian cells. The envelope protein E2 binds to the cell surface via an unknown receptor and possibly glycosaminoglycans as the attachment factors. The Chikungunya virus enters a cell through clathrin-mediated endocytosis. Acidification of the endosome leads to the insertion of the fusion peptide in E1 into the endosomal membrane. Fusion of the viral envelope and the endosomal membrane releases the nucleocapsid into the cytosol. Disassembly of the nucleocapsid releases the positive sense genomic RNA and the non-structural protein translation occurs. Four non-structural proteins together with genomic RNA and presumably host proteins assemble at the plasma membrane and modify it to form viral replication compartments containing viral double-stranded RNA. Non-structural protein P1 to P4 function as a replicase and localize to the spirule neck to generate genomic, antigenomic, and subgenomic viral RNAs. Spirule internalization allows the formation of large cytopathic vacuoles that house multiple spirules. Spirules at the plasma membrane or within the cytopathic vacuole 1 are fully functional. Translation of subgenomic RNA produces the structural polyprotein and capsid autoproteolysis releases free capsid into the cytoplasm. Translocation of envelope proteins E3, E2, 6K, E1/E2, E2TK polyproteins into the endoplasmic reticulum. E2 and E1 are post-translationally modified and transit the secretory system and are deposited at the plasma membrane. Interaction of the capsid and the genomic RNA leads to the formation of icosahedral nucleocapsids. The nucleocapsids assembly with the E2 and E1 at the plasma membrane results in the budding of mature progeny virions. Later in infection, cytopathic vacuoles 2 form containing hexagonal lattices of E2/E1 and are studded with nucleocapsids. Cytopathic vacuoles 2 likely serve as transport vehicles and assembly sites for structural proteins, allowing the formation of mature virions and egress. Pathogenesis of Chikungunya virus. Acute chikungunya infection begins with the transmission of the virus via a bite of infected mosquito to the skin, where it replicates in susceptible cells, including fibroblasts and macrophages. The virus disseminates through the lymphatics and bloodstream to typical and atypical sites of primary replication. Acute infection elicits an inflammatory response in infected tissues, characterized by an extensive infiltration of mainly macrophages and monocytes, but also neutrophils, natural killer cells, and lymphocytes in target tissues, and by elaboration of a number of pro-inflammatory chemokines and cytokines. Within arthroskeletal tissues, synovial hyperplasia begins. Synovial hyperplasia refers to an increase in cellularity of the synovial membrane and it leads to thickening of the synovial areas. Synovial joint is the type of joint found between two bones that move against each other. Characteristically, it has a joint cavity filled with fluid. Viral replication and host immune responses can cause myalgia and polyarthralgia in distal joints. Myalgia refers to a pain in a muscle or group of muscles, and polyarthralgia refers to pain in several joints. Chronic chikungunya viral disease can persist for months or years after acute infection, but it is often limited to more distal joints. Chronic disease is likely mediated by persistent virus and inflammation. Possible sites of chikungunya virus persistence include endothelial cells in the liver and other organs and mononuclear cells in the spleen, macrophages within the synovial fluid and surrounding tissues, and satellite cells within the muscle. Within the chronically infected joint, the continued presence of a subset of infiltrating cells, mainly macrophages, monocytes, and lymphocytes, and specific pro-inflammatory mediators like interleukin 6, interleukin 8, and MCP1 within the synovial fluid likely contribute to the prolonged inflammatory disease. Chronic joint pathology resembles that in rheumatoid arthritis with significant hyperplasia and angiogenesis. Symptoms of chikungunya virus. Chikungunya symptoms can vary from person to person but can include fever, severe joint pain, rash, significant swelling of joints, muscle pain, and headache. Symptoms usually show up within 3 days or a week of being bitten. In most cases, patient will recover in a week from the symptoms, although recovery times can last months or even years in some cases. Now, let us discuss the applicability of different diagnostic methods in the course of chikungunya viral infection. In acute phase, viremia can persist up to 5 to 7 days post-infection onset, and chikungunya viral genomic RNA can be detected by RT-PCR reliably until 7 days post-illness onset. It is therefore suggested that the detection of chikungunya viral RNA and virus isolation from serum samples for diagnostic purposes should be done before 5 days post-illness onset, because the chance of false negative results increases with the decrease in viral load. IgM and IgG antibodies against chikungunya virus begin to be produced 2 days post-illness onset and 4 days post-illness onset, respectively. Stable titers of IgM can be seen in serum from day 6 post-illness onset till around 4 months post-illness onset, and can be detected mostly until 6 months post-illness onset, whereas sustained levels of IgG can be present for more than a year. The antibodies against chikungunya virus can be detected by immunoassays after the development of humoral immune response. In case of IgG, long into the chronic phase, both symptomatic or asymptomatic. Now let us look into the preventative measures to control the spread of chikungunya viral infections. Use insect repellents that has DEET or picaridin. This will help us prevent the multiplication of the vectors of the viral infection. Do not allow water to stagnate. Use mosquito nets while sleeping. Protect rooms by using mosquito repellent screens. Avoid social gathering in outbreak areas. Use mosquito coils. Wear long sleeve shirts and pants when traveling to mosquito infested areas. Avoid traveling to places of chikungunya outbreak. Now let us look into the vaccines available for chikungunya. Although chikungunya is endemic in many regions, at the moment, no vaccines are available, and the treatment of chikungunya infected patients is mainly based on the use of anti-inflammatory drugs for symptomatic relief. Importantly, a chikungunya vaccine needs to be economical, easy to handle, and easy to transport, thus enabling developing countries to be able to afford a mass vaccination campaign. At present, there are several clinical vaccines in development, and a limited number of clinical trials are currently being carried out across the globe. Chikungunya preclinical candidate vaccines under preclinical development are diverse. This is a summary of all current vaccines under clinical trial evaluation, either phase 1, phase 2 or phase 3. VLA1553 is a live attenuated vaccine developed by Valneva, Austria. It is given as a single intramuscular dose. It is under phase 3 clinical studies. The PXVX0317 vaccine, developed by the US National Institutes of Health and Paxvax, is also a live attenuated vaccine, which is also given as an intramuscular dose, is currently under phase 3 clinical study. It needs to be injected in 2 doses to be effective. The next vaccine is the MV-Chick202 vaccine, which is also a live attenuated vaccine developed by Themis Bioscience. It is under phase 2 clinical study. The BBV87 vaccine is a inactivated whole virus vaccine, which is developed by International Vaccine Institute and Bharat Biotech International. The CHAdOX1 chick vaccine is an adenoviral vector-based vaccine developed by the University of Oxford. The mRNA 1388 vaccine is an mRNA-based vaccine developed by Moderna Technologies and is currently in phase 1 clinical trial. The HB1CG1 2020 vaccine is a whole cell inactivated vaccine developed by Human Biological Institute, India. It is currently under phase 1 clinical trials. No specific antiviral treatment is available for chikungunya. Treatment for symptoms can include rest, fluids, and use of analgesics and antipyretics. For patients with persistent joint pain, use of non-steroidal anti-inflammatory drugs, corticosteroids, including topical preparations and physical therapy may help lessen the symptoms. Now, let us look into a brief summary of chikungunya disease. Chikungunya is a disease transmitted by the bite of Aedes aegypti and Aedes albopictus mosquitoes. The main signs and symptoms are high fever, which can last up to 7 days, and joint pain. Other common clinical manifestations are headache, itching, rash, muscle pain. What should one do if they observe these signs and symptoms? They should seek medical attention. Do not self-medicate. They should drink enough fluids. It is recommended to drink oral rehydration solutions. If patients are breastfeeding, they can continue to do so. These are a few of the references for further reading. Let us discuss some multiple choice questions on the chikungunya virus. Which type of viral RNA genome does chikungunya virus possess? A. Single-stranded positive sense RNA. B. Double-stranded negative sense RNA. C. Double-stranded RNA. D. None of the above. The correct answer is A. Single-stranded positive sense RNA. How is chikungunya virus primarily transmitted to humans? A. Through contaminated water. B. By mosquito bites. C. Through sexual contact. D. By airborne droplets. The answer is B. By mosquito bites. What is the main approach to treating chikungunya virus infection? A. Antibiotics. B. Antiviral drugs. C. Supportive care to relieve symptoms. D. Vaccination. Answer is C. Supportive care to relieve symptoms. Which of the following is not a preventive measure against chikungunya virus? A. Using insect repellents. B. Eliminating mosquito breeding sites. C. Taking antibiotics prophylactically. D. Wearing long sleeved clothing. Answer is C. Taking antibiotics prophylactically. Which symptom is not typically associated with chikungunya fever? A. Fever. B. Joint pain. C. Skin rash. D. Persistent cough. Answer is D. Persistent cough. Thank you for your kind attention. Protect yourself from mosquito bites.